Hi Year 12s, this is going to be our last video for cells and also the last PowerPoint we'll cover in class for cells. Once we've done this slide, you've completely finished the second topic of the Stage 2 curriculum for biology SACE. In this PowerPoint we're going to be looking at cell culturing, which will talk about how we actually do cell culturing, the advantages and disadvantages of cell culturing, as well as some talking about reproductive cloning. Let's begin. So you'll notice first that the need to know here is quite small. As I said before, we're getting to the end of the curriculum, so only 2 need to know here. If you'd like to write them down, if you're keeping a record of these, make sure that you pause the video and write them down. Firstly, what exactly is cell culturing? Well, cell culturing is the process of growing cells outside of the body of a living organism. We as a human race, we use cell cultures we use cell culturing to grow cells from microorganisms, but occasionally we do also grow cells from animals and plants as well. The technique that we use for cell culturing, there's 3 main things we need to discuss. We need to dissection, conditions for growth, as well as the cell culturing itself. So firstly, dissecting. So the first stage in any cell culture that we need to do is we need to remove the tissue or the cells from the source that we're going to be using. We do this through either chemical or physical means, but know that always the first stage in cell culturing is the dissection or the removal of tissue from the source that you're going to be growing the cells from. Next, you need to make sure that conditions for growth in the laboratory are perfect. So you need to make sure that you've got nutrients to allow the cells to grow at the correct levels. You need to have oxygen for aerobic respiration, unless you're going to be looking at something like yeast or alcohol production, etc., where you're going to want anaerobic respiration, but that's not important right now. You're going to need to make sure you've got growth factors such as hormones to allow the cells to grow. You need to have osmotic balance to ensure that the cells aren't going to go into hypertonic or hypotonic modes. You need to have optimized pH and temperature for the type of cell that you're using, remembering that every cell has their own optimal pH and temperature. And lastly, you need to have antibiotics, because anytime you're using cells in a laboratory environment outside of the human body, there's always the chance of contamination. So you put antibiotics there to allow that bacteria to be killed off or prevent them from actually getting into the cells. Lastly is the cell culturing process itself, where the cells will be transferred to a suitable vessel and they'll be incubated at an optimum temperature and allowed to grow. But what do we actually use cell culturing for? So it's fantastic we've been able to grow these cells in a laboratory environment. What are we going to use them for? So there's 5 main things that we're going to talk about. So the first one's research. So by using cell culturing, we're able to study cell structure and function outside of the human body. So we can actually do that in a laboratory setting. Toxicology, we can, all the time, medicine's trying to create new drugs to fight off different diseases, etc. By having cell culturing, we can see the effect of these new drugs without actually testing them on humans first. So the first test would be to test them on cell culturing to see what's going to happen to them. Cancer research, we can use the cell cultures to improve radiotherapy and chemotherapy and hopefully reduce the amount of cell death from those processes. Research virology, we can study the interactions between viruses and cells, again outside of the body rather than putting human life at risk. And lastly, genetic engineering. We can genetically engineer cells for our own purposes and see that the effect of those modified cells will actually have. Of course, whenever you're playing God as such and creating your own cells, there is going to be advantages and limitations. So the advantages are that we can perfectly control the environment. It's not like the human body that may change at certain times or animal bodies that may change. We can make sure the laboratory setting has exactly the right conditions for those cells to grow. We can successfully use them to study interactions between medicines and different cell types. Fantastic. Cell culturing is used by medical scientists frequently to diagnose diseases from tissue samples taken from humans. We've created many new antibiotics from the processes of cell culturing, and cultured cells can be preserved and kept for later on and studied various times. Great example of that is HeLa cells. We won't talk them in depth in this PowerPoint or video, but if you'd like to know them, search up HeLa cells, H-E-L-A, HeLa cells, and that will be a great research point. Of course, there are disadvantages. So whenever you're growing something like this in a laboratory setting, there's always the chance of contamination by microbes, which could cause quite a few problems. Sometimes when you're studying the effects of cells outside of the human body, outside of their normal conditions, you will not be able to identify all of the effects a drug may have. So when you use a pathogen or a drug inside the body, there may be various side effects that you didn't actually see in the cell culturing process. That's why all drugs, etc., go through the process of human trials before they're pushed. So you'd go cell culturing, human trials, then released to the public. Obviously, anytime you're growing cells rapidly, you're going beyond the normal growth rate, you're going to have the chance of introducing mutations. There is always the chance as well, and sometimes this is always the case, that protein expression is very different in cultured cells because of the absence of other cells in the body. So protein expression actually changes. And lastly, that non-cancerous cells, which are the cells we generally want to use because cancerous cells may act differently, non-cancerous cells have a finite capacity for growth by cell division. So they can only divide so many times before that cell is finished. So we're constantly having to do and get more cells. Again, though, a great point to talk about there would be your HeLa cells. So I would encourage you to do some research on HeLa cells.
And I'm getting quite near the end now of this analysis.
Lastly, we're going to look at reproductive cloning.
What the? It made another me!
Reproductive cloning is a technique used in genetic engineering to clone organisms such as plants and animals. Obviously, the most famous one that we know about is Dolly the sheep. Don't need to know about Dolly the sheep in detail, but if you do want to know a little bit more about it, there is obviously this PowerPoint here, or this little video / picture here, that will show you exactly how Dolly was created. The main reason that we do reproductive cloning, this is what you need to know, is generally just to increase the populations of rare and endangered species. And just be careful that I have written rate there instead of rare, but that should be increase the populations of rare and endangered.
That's all, folks.